All right, now we're going to start getting into the details of the three stages of aerobic cellular respiration, which is cells using oxygen to break down food. It's what we call burning our food, to release the energy from the food molecules, and then using that energy to build ATP so the energy can be stored in ATP. We're going to go through each of these three stages in a lot of detail, and I'll probably devote a separate screencast video to each stage. So there'll be a video for glycolysis, another one for Krebs, another one for oxidative phosphorylation. But we're going to start with glycolysis, the breaking down of food out here in the cytosol. And the food we're going to focus on is sugar, glucose. So we're going to look at the breakdown of a glucose molecule through this aerobic respiration process. And if this looks like a very complex slide, it's because it is. But as I said, we're going to be spending essentially the entire video just going through this slide and this process. So the first stage of aerobic respiration of glucose is going to be glycolysis. And the word glycolysis, glyco, is Latin for sugar. Glyco is the old Latin word for sugar. Lysis, we already talked about the word lysis when we talked about osmosis. That means to break in Latin. So glycolysis or glycolysis, that means breaking sugar. So what we're going to do here in this first stage is we're going to break sugar. We're essentially going to break a sugar molecule in half. But here's where it gets interesting or difficult, depending on your perspective. We want to think about sugar and where in the sugar molecule is the most energy. We're going to have to release the energy from sugar. But how are we going to get that energy out of sugar? Well, to get energy out of food, you have to break bonds in the food. The energy is stored in the electrons that are in the shared orbits in the covalent bonds. So glucose is a molecule made up of carbon atoms, hydrogen atoms, oxygen atoms, but those atoms are bonded together covalently, which means electrons are being shared in common orbits between the atoms. So in order to get the energy out of this system, of this chemical, we're going to have to break some of those bonds. In fact, we're going to end up breaking most of the bonds that are holding the glucose molecule together. Now, when we break the bonds, we actually break apart the orbits that the electrons are sharing. So we might have two electrons spinning in a common orbit, but when we separate those atoms and break the bonds, then one electron goes back with one atom and the other electron goes back with the other atom. Now, in order to really get energy to come out of the system, we have to not just separate the atoms. That just makes unhappy atoms usually and doesn't necessarily give you energy. What you need to do is you need to slow the electrons down. Most of the energy in a atom that's accessible is the energy that's powering the electron orbits, the electrons to spin around the nuclei of atoms. So the energy that's in the motion of the electron, that's making the electron spin real fast, that's the energy we can harness. But in order to get that energy out, we have to slow down the electrons. So when electrons are going very fast, which they're usually doing when they're in a covalent bond, then they contain a lot of energy. It's the energy that's moving them real fast. But when we slow those, when we break the bond and then slow the electrons down, put them in a new bond where they don't have to go as fast, then the energy that was making them go fast, some of that energy is released because now they're moving slower. So let's see if I've got something I can use to illustrate that. All right, so a quick animation here. Here's an electron orbiting a nucleus, where the blue is an electron here. And what I'm going to do is I'm going to slow it down. Okay. So if I can slow that electron down, then it will give up energy, because it takes less energy to move things slow than it does to move things fast. It takes more energy to move something faster. So the electrons that are in all these bonds in sugar are moving very quickly. When we break the bonds and in sugar and create smaller molecules from the sugar, the electrons that are in the smaller molecules don't have to move as quickly. They move more slowly. When the electrons slow down, they give up some of their energy. We can use that energy that's given up by those electrons to push together ADP molecules with phosphates to make ATP molecules. Remember, that's the energy storage chemical. So here in glycolysis, we're taking glucose molecules, we're breaking glucose in half into two smaller molecules. The electrons can move more slowly in the bonds in these smaller molecules than it moves in the glucose. So a little bit of energy is given up here, enough to push together two ATPs and build a couple of ATPs. But there's still a lot of energy left in these smaller molecules. And the atoms that have the most energy, because they have the fastest moving electrons, are the smallest atoms. And the smallest atoms in sugar and other food molecules are hydrogen. Remember, that's the littlest atom on the periodic table. Hydrogen atoms, because they're the littlest atoms, actually have the fastest moving electrons. So those are the atoms where we can get the most energy by slowing down their electrons. So during the process of cell respiration, particularly during the first two stages, we're going to be removing hydrogen atoms from the sugar molecule. As we break down the sugar, we're going to remove the hydrogen atoms. Removing the hydrogen atoms from sugar helps us break down the sugar, and also gives us the real high energy electrons. Now, how are we going to remove hydrogen from food? Well, if we just pull off the hydrogen atoms, we're going to create a lot of unhappy atoms. And remember what unhappy atoms want to do. They want to bond to something else so they can get happy again. So when we pull hydrogen atoms off of glucose, for example, hydrogen with high energy electrons, we've got to attach those hydrogens to something else. And in this case, we're going to attach those hydrogens to a hydrogen carrier molecule called NAD. We're going to talk more about how NAD works. So NAD molecules are molecules that can bond to free hydrogen atoms. And when they do, they become NADH. That's the NAD with an attached hydrogen atom. So here with these NADs, we're giving the free hydrogen that we've pulled off of glucose something to bond to, because hydrogen by itself is a very unhappy atom. It's going to need to bond. So we're going to give it this chemical NAD to bond to, and when the hydrogen bonds to it, it becomes NADH. So in this first stage, glycolysis, we remove a couple of hydrogens from glucose. We transfer those hydrogens to another chemical called NAD, which becomes NADH. NADH doesn't just accept the hydrogens, it can also move the hydrogens. NADH is a hydrogen carrier molecule. So it doesn't just grab the hydrogens from sugar, it then moves those hydrogens, it transports into the mitochondria, specifically to the inner membrane of the mitochondria where the hydrogens are going to be dropped off. So NAD is a hydrogen transporter molecule, taking hydrogen atoms from food, because these are the high energy atoms, and then moving them into the mitochondria where those hydrogens are then going to be released so that their electrons can be slowed down. In the process of removing hydrogen from glucose, we break apart the glucose molecule and it becomes two smaller molecules called pyruvic acid. Acids are what you get when you take hydrogen atoms off of sugars. So we get two smaller acid molecules, pyruvic acids. There is enough energy lost from the glucose just in breaking it in half and removing a couple of hydrogens that we can use. There's some extra energy released here, and we can use that energy to push together a couple of ADPs with phosphates and make a couple of ATPs. So here in glycolysis, we remove 2 hydrogens and we use the energy that's released to build 2 ATPs. And we get 2 molecules of pyruvic acid. That's what the glucose becomes when it breaks apart. Interestingly, the mitochondria here cannot absorb glucose. Glucose cannot get through the outer membrane of the mitochondria or the inner membrane. So that's why mitochondria can't break down glucose completely. They need it broken down partially first because glucose can't get into the mitochondria, but pyruvic acid can, and NADH can. So here in glycolysis, we're turning the sugar into a couple of chemicals that actually can get into the mitochondria. NADH, which is carrying the hydrogen, and pyruvic acid. Pyruvic acids actually still have a lot of hydrogen atoms attached to them, so there's a lot more energy we can get out of them. And we're going to get that additional energy out of them here in the matrix. When we do the Krebs cycle, that's where we're going to remove the remaining hydrogens from pyruvic acid. The hydrogens that were removed already here and transferred to NAD, which became NADH, those are going to be dropped off in the inner membrane in the third part of cell respiration. So if we look at this as just chemistry, we can summarize all this as a chemical reaction where we take sugar molecule, we use 2 NADs and bring in 2 NADs in order to take the hydrogen off of sugar. Once the NADs pick up the hydrogen, they become NADH. Once the sugar loses its hydrogen, it falls apart into a couple of acid molecules. And then there's some energy released in that process, enough energy to put together 2 ADPs and 2 phosphates to make 2 ATPs. So if you just look at this as chemistry, there's a couple of sets of reactions that describe this entire process. Now I'm going to link in a video for you that shows you the function of NAD, because NAD is something, hydrogen carriers are something we haven't talked about yet. I can also use this little program here. You can think of NAD as a hydrogen truck. It's essentially just like a regular truck. It picks stuff up and then brings it somewhere else and then drops it off. So NAD is a hydrogen truck, and food molecules have hydrogen atoms. The hydrogen atoms have the fastest moving electrons, so they have the most energy. And what NAD does is it removes a hydrogen atom from food, and then when it does that, it becomes NADH, and the food molecule is now missing hydrogen, and it starts to fall apart. So this is the process that gets food to start to fall apart, is when you remove the hydrogen from it. The bonds are broken, now they have to rearrange in a new way. And then the NADH here, which is created, can bring that hydrogen to another part of the cell where the hydrogen electrons can be slowed down and give up even more energy. So that's the basic process of just glycolysis. This is just the first stage of cell respiration. And I know this is complicated. That's why I'm linking in professional videos. So there's a professional video in NAD right after this screencast, and then there'll be more later on to explain other details of this process.